Let's look at topic 2.6, membrane transport. Your overall enduring understanding on this topic is that cells have membranes that allow them to establish and maintain their internal environments that are going to be different from their external environments. So when we refer to this topic, the main thing we're going to talk about is transport, which is the movement of substances into and out of the cell. Now, within our membrane transport, we are going to see some patterns. These are going to be the two things that will decide how the molecule is going to move across the membrane. Okay, so when we're looking at the patterns of membrane transport, this is the ability of a molecule to move across the membrane. Okay, so the first of these is going to be the semi-permeability of our plasma membrane. Okay, so depending on that semi-permeability and the molecule, we'll see how the molecule decides to move. The second is going to be the size and the charge of the particle. Okay, so the size of the molecule is going to determine the mechanism that's being used to move that molecule in and out, and same thing for the charge of the particle. We have two types of membrane transport we're going to focus on. We've got passive and active. With our passive transport, this is going to have no outside energy involved. Okay, so this is passive. No outside energy is required in order for these to occur, and active, on the other hand, does require energy. With our passive transport, it is important for us to remember one of the more essential parts that, or essential roles that passive transport plays, is that it helps cells import and export their resources. So cells are able to import those resources that they need, and they can export their waste without having to use any of their energy. Now, within our passive transport, we are going to see that molecules are going to move from an area of high concentration to an area of low concentration. Oftentimes, it helps to remember passive goes with the flow. So we're going from high to low. So this is actually one of the more important things you guys have to remember with it, is that with our concentration gradient, passive is going from high concentration to low concentration. Now, the first type of passive transport we are going to look at is going to be related to the structure of the membrane. We know that our membrane is structured with our phospholipid bilayers. The phospholipid bilayers create that hydrophobic sandwich within our membrane. That means that the inside of our membrane is hydrophobic. We have a concept called like dissolves like. So because the inside of the membrane is hydrophobic, it would be easier for other hydrophobic particles to go through. So small lipid soluble hydrophobic substances are going to be able to cross through the membrane without any resistance. Okay, so this process is called simple diffusion. Now, when we're looking at our diffusion, if we're referring to gas particles, we are calling it diffusion. But another component is going to be, if those molecules we're referring to are water molecules, this will be called osmosis. And we're actually going to go into osmosis later on in this unit. Now, simple diffusion, again, these are your hydrophobic or small non-polar molecules. They can just kind of drift right through our membrane because they are similar to the inside of the membrane in structure. So we will not have any protein channels, and there will be no energy required. An example of simple diffusion is going to be seen in our kidneys. We'll see in the kidneys that there are solutes from the blood that are diffused into the Bowman's capsule of the nephron. Okay, so this is an example of simple diffusion within our bodies. All right, let's go on to the next type of passive transport. So the next type of passive transport we're going to look at are these two channels that you see. Okay, so we'll have the use of channels, so this is called facilitated diffusion. Okay, within facilitated diffusion, we're going to see these are going to be our hydrophilic substances. So hydrophilic substances are not going to want to move between that hydrophobic interior because they don't want to have that contact with that hydrophobic interior. They are going to be using our channels. Okay, so this is where our proteins come in because these hydrophilic substances need assistance, and that assistance for them comes in the form of protein channels. There are a number of protein channels that are used by cells to pass hydrophilic or polar substances through that are going to be acting as tunnels. Now, when we refer to these channels, these are specialized types of channels or specialized types of tunnels that will make sure that they only let certain substances through. Okay, so they are going to be very specific to only allow certain molecules through into the cell or out of the cell. Okay, so this is another important component is that we do have a lot of variety in our channels. Okay, an example would be aquaporins. Aquaporins would be channels that are water specific channels, so they would only allow water molecules to go into or out of the cell. So they don't allow just anything through. It's not open to just any hydrophilic substance. They are very specific. Okay, so facilitated diffusion, you have to know this does need the help of a protein, but we still don't have any energy required for facilitated diffusion because we are going from that high concentration to the low concentration. Now let's take a look at our proteins. So as you guys can see on here, we have two different types of proteins that are used in facilitated diffusion. We have the one on the left, which would be known as our channel proteins. So and then we have our right, which is our carrier protein. Now, each of these has their own unique function and is used in its own capacity. Our channel proteins, these are generally going to be only water or certain ions. Okay, so these channel proteins are used only with water or certain ions. Okay, so they allow it to channel through without coming in contact with the hydrophobic interior. And it is always going to be from that higher to lower concentration if we are looking at the facilitated diffusion. Now, carrier proteins are somewhat different. As you can see, the carrier protein, it has that specific shape. Okay, so carrier proteins are going to change their shape to carry the substances or specific molecules across the membrane. Okay, so these are even more specific because they'll change their shape. Now let's take a look at active transport. As we know, active transport requires energy, So we will see that ATP is going to be involved with active transport. The difference coming from our passive to our active is that in active, we're going from an area of low concentration to high concentration. Okay, so because we're going from that low concentration up to the high concentration, we are move this is going to be movement that's going against that natural flow that we saw in passive transport. And now because we are moving against that natural flow, that's why these require energy, which is our most commonly going to be ATP. Now, in active transport, the transport is going to be helped by transport proteins. Okay, so that right there is an example of a transport protein. These are going to be the proteins that are powered by ATP. So the ATP is actually coming in with the proteins. So an example, the most common example that we are going to see and you're going to be looking into a lot is the sodium potassium pump. And we're going to go into more details on this pump later, but in general, the role of this pump is to pump out 3 sodium ions, pump in 2 potassium ions. And in order to do that, it depends on ATP to get the ions across the membrane that otherwise would have just stayed in the area of higher concentration. Okay, so it's moving them against that natural flow. So it's letting them go from low to high concentration. And because it's making them flow in that opposite flow, it needs ATP. Within active, we're going to see that we do have primary active transport and secondary active transport. And the key difference is that the ATP in primary is ATP that's directly utilized to transport our molecules across the membrane, whereas in secondary, the molecules that are actively being transported across the membrane are using it's using energy that was gathered by the cell from the movement of the particles that were moving along the gradient. So another substance within the cell that moved or flowed down its concentration gradient. So what we saw in simple diffusion and facilitated diffusion. So the energy that was gathered from that, when that energy is being used, that would be our secondary active transport. Now let's take a look at a few other types of active transport. So a few of the types that we're going to see for active transport is going to be bulk transport. So bulk transport is a one-way movement. Kind of sounds just like the name, bulk. And it's that one-way movement of fluids, and it's really powered by pressure. So for example, blood in your body, the blood flow is an example of bulk transport, or the movement of fluids in the xylem of plants is an example of bulk transport. And so this does require some energy, and so it does fall within active transport. Other two examples would be endocytosis and exocytosis. Let's look at exocytosis first, because this is just the ability to eject waste or specific secretions, such as hormones that, or secretion products like hormones, out of the cell. Okay, so in exocytosis, that word exo, use that to remember exit. So these are going to be things that are exiting our cell. So if we'll see our molecules, I'll form a vesicle around them and then just kind of push them out of our cell. Endocytosis is going to be the opposite of exocytosis. Okay, so endo, it is entering. Okay, so this is where our substances are going to be entering. Now, endocytosis will come in whenever the particles that need to enter the cell, if those particles are too large, that they're not able to go through our channel proteins or our carrier proteins or transport proteins, then the cell is going to use endocytosis, where it takes a portion of the cell membrane to engulf that particle or that substance. Okay, so the overall process we'll see with these is that we'll have a pocket being formed, then it pinches in and forms a vesicle or a vacuole, depending on what the substance is, and ingests that substance. Okay, so here you'll see that there's that substance that we need to bring into the cell. So our cell's forming that pocket. That pocket then pinches into the cell to bring it in, and then that pocket, once it's pinched in, it's within that vesicle or the vacuole inside of the cell. Now, endocytosis has three different types. Okay, so we do have three types of endocytosis, and it mainly depends on the process that's being used or the substance. The first would be pinocytosis. This is when we're ingesting liquids, so this is known as cell drinking. Our second type is phagocytosis. This is where the substance that's involved is going to be a solid. Okay, so this is referred to as cell eating. Our third type, these are receptor mediated endocytosis. Okay, so this is a more specialized version of endocytosis. We are going to see that we've got some cell surface receptors that are working within the endocytic pits. And those pits, on the inner part of the pits, we are going to see a specific protein that is known as clathrin. Okay, so we've got our receptors, and then we have some proteins on the inside that are known as clathrin. Now, when our receptors are going to be filled with the specific substance, we're going to then see that the ligand is going to be brought into the cell by a process of folding into the cell membrane. Okay, so you'll see right here that these receptors are specific to that triangle shaped substance. So once those triangle shaped substances have filled each of our receptors, so once all three receptors have been filled, we'll see a little process of folding in, and it'll fold those substances into the cell, and that would be our receptor mediated endocytosis. Okay. So let's come back to our essential knowledge list from our College Board CED. So it's just to kind of double cross everything that we wrote down in our notes. So one of the first things that we see in our essential knowledge is that you have to understand that passive transport is the net movement of molecules from high concentration to low concentration without the direct input of metabolic energy. So when we took our notes, we looked at that we have passive transport, we're going with the flow, we're going from high concentration to low concentration, and there is no energy required. Next on our list is understanding that passive transport is going to play a primary role in the import of materials and the export of waste. And as you can see, we have that in our notes, that this is essential because it helps cells import the needed materials and export their waste without having an input of energy. Next on our list, we see that we need to understand that active transport will require the direct input of energy to move molecules from regions of low concentrations to regions of high concentration. So we come down to our notes, we have that listed, the active requires energy, and that we're going from areas of low concentration to areas of high concentration. Next up on our essential knowledge, we need to understand the next part right here. The selective permeability of membranes allows for the formation of concentration gradients of solutes across the membrane. 2.E.1. So this is more of an understanding of that concept. You need to understand that your membrane is selectively permeable, meaning only certain things are allowed in and out. Okay, so when we're doing that, when we are restricting what's going into the cell and what's going out of the cell, that is allowing us to form a concentration gradient. So that gradient of having low concentration on one end and high concentration on the other end. That is only possible when we have that selective permeability. Okay, if everything was allowed into the cell and could get into the cell whenever they wanted, however they wanted, we would not develop those concentration gradients. We're only developing those concentration gradients because of that selective permeability. Now, what a concentration gradient, we are actually going to go further into that when we go into topic 2.7. So the next part is understanding how larger molecules are moved into and out of the cell, and that brings us down to endocytosis and exocytosis. And when we looked at endocytosis and exocytosis in our notes. Next, we're going into topic 2.7, facilitated diffusion. Now, a lot of this actually is stuff we've already talked about in 2.6. So we're going to kind of go through this a little bit more quicker. First thing that we have in our essential knowledge is that membrane proteins are required for facilitated diffusion of charged and large polar molecules through a membrane. And the first part of that is that large quantities of water will pass through aquaporins, which we looked at in 2.6. Remember that aquaporins, these are your water channels, and water can flow faster if it's going through these channels rather than it depending on diffusing through the hydrophobic or phospholipid bilayer. So instead of going through that hydrophobic interior in small quantities, aquaporins allow it to go through much faster. The next part is that charged ions, including sodium and potassium, require channel proteins to move through the membrane. What exactly are charged ions? So an ion is going to be any of your atoms or molecules that have a charge. We looked at this when we reviewed our chemistry. So the charge of +1 charge on sodium makes it a charged ion because it lost an electron, has a charge of +1. Same thing with potassium, that +1 charge makes it an ion. So you have to understand that our ions cannot go through that hydrophobic interior. They do require channel proteins. Next thing you need to remember is that the membranes may become polarized by the movement of ions across the membrane. What exactly does this mean? So polarized membrane is that result of anytime our membranes are moving or storing different types of ions on different sides of the membrane. Okay, so because we're taking these charged particles and we're either moving them across or we're, taking certain charges, putting them on one side, leaving other charges on the other side, this ends up causing the membrane to have one side of the membrane having developed positive charge and the other side having a negative charge. And then we would call it a polarized membrane. The other essential knowledge that we need to know is that membrane proteins are necessary for active transport. We looked at that in 2.6, so we're going to go on to the next one, which says that metabolic energy such as ATP is required for active transport of molecules and or ions across the membrane and to establish and maintain concentration gradients. So we understand the part about energy being required in active transport. But now what we're going to look at is how exactly is this contributing to establishing and maintaining concentration gradients. So let's first go into our concentration gradients and look at what concentration gradients are. So a concentration gradient is going to be that process of our particles moving from one side of our membrane to another. So they're moving from an area of higher concentration to an area of lower concentration. Okay, so this is that concentration gradient. And we looked at that throughout 2.6 in our passive transport when we were going from lower to higher concentration. So you have to really understand on this gradients. Is that with active transport, we're using that ATP and moving our ions across the membrane, and that entire process when we are moving our ions opposite the flow. So when we're taking our ions and we're moving them from our lower to higher concentration, we're actually helping to maintain and establish that concentration gradient. Even though active transport in itself is moving molecules in the opposite direction, but that process of active transport, moving them in the opposite direction, ends up balancing out what's happening naturally within the environment, and it helps to establish and maintain that concentration gradient for our cells. The last essential knowledge that we see in 2.7 is that sodium-potassium pump and how it contributes to the membrane potential. So the sodium-potassium pump, if we come down here, we'll see that we have our 3 sodium molecules, and we'll take a part of our ATP, and the phosphate group we're taking from the ATP is going to allow our pump to change its structure. Okay, so that phosphate group that it's going to take is going to allow it to change its shape and then basically restructure itself to where it will now open up to the outside of our membrane. Okay, so now it's going to be facing out, and it lets those 3 sodium ions and it sends them out. So we've sent 3 positive sodium ions out, and then we'll take 2 potassium ions and we'll take them in. Okay, so then we'll take another component of our ATP and basically do that same process again. We're going to change shape again and change our structure to now face in, letting those potassium ions into the cell. Okay, so we looked at what sodium, potassium, and the overall view of this in 2.6 and how it takes 3 sodium, pumps them out, takes 2 potassium, pumps them in. So the sodium potassium pump, now in 2.7, we have to understand that it plays a big role in maintaining membrane potential for our cells. Okay, so it allows our cells to maintain a higher concentration of sodium outside and a higher concentration of potassium on the inside. And by doing that, and through that loss of our positive ions, we are actually stabilizing the cell's membrane potential. Okay, so making the cells maintain that negative resting potential inside. Now, when we say negative resting potential, so you look at what happened. We have 3 positives that went out, 2 positives came in. If you look at that overall charge, that means that the inside now has a negative 1 as an overall charge. So now it's allowing those cells to maintain that negative resting potential on the inside. Okay, so this is actually essential to a lot of cells, and it's essential to help them perform their functions. Okay, so that loss of ions from the sodium potassium pump is stabilizing the cell's membrane potential so that cells can do the functions that they're supposed to do. All right, so that is it for 2.7.